My name's Elizabeth Fichtner, and I'm a farm advisor. I cover walnuts in Tulare and Kings County. And I chose to talk about walnut scale today and join the cadre of entomologists because we've seen a tremendous amount of walnut scale building up throughout the whole Central Valley and including my region in the southern San Joaquin. At the end of the season, or towards the end of the season, we saw a lot of really bad looking walnut orchards, a tremendous amount of thinning in the upper canopy and flagging. And this was true in very productive orchards and also declining orchards. It was, across the board. And when I went on farm calls, one of the clear things that I saw that seemed very consistent was a tremendous load of walnut scale. And historically, a long time ago, walnut scale wasn't considered to be a very important pest of walnuts, but something has changed, and this organism, when it's at high populations, it can cause a tremendous amount of damage. One key to the amount of damage that it causes is that it has been found to be in association with the Botrytis fungi, which cause these canker diseases on walnut. It appears to have both a direct association. You see the photo in the bottom right. You get these necrotic islands directly underneath the scales. But it also appears to predispose the new tissue, like in this photo, to scale infestation, where you have the older scale down here on the older wood tissue. And this is the Botrytis canker on the new tissue that's not even colonized by those adult scales yet. So there's both a direct association and an indirect association. And Dr. Themis Michaelides has done extensive work on this phenomena. I'm just going to present one of his slides on the topic. When his team inoculated twigs with a spore suspension, which kind of mimics what would happen in the natural environment, they inoculated with these 3 different pathogens that cause canker disease on walnut. Twigs that had scale on them had significantly higher levels of disease incidence than those that were not infested with the scale. So the dark colored bars are the twigs that were infected with scale. So there was 60 to 75% more infected shoots with scale than those without scale. So there's a clear situation where the scale has predisposed these twigs or branches to infection with these Botrytis fungi. Now, walnut scale is not the only scale that's found on walnut. So I want to briefly cover what else is out there so that you can differentiate between these when you look in your own orchard. We often group frosted scale and the European fruit lucanium together. And why is that? They're actually the same genus. And we group them together because you basically can't tell them apart except for one stage in the spring, which was around March and April, where the frosted scale will exhibit this waxy coating. But otherwise, the immature stages look similar. There's also the Italian pear scale. It's a more reddish color, and it tends to be associated with lichens and moss. This is not a major pest on walnut. And then we have San Jose scale and walnut scale. Of course, my talk is focused on walnut scale, but its closest look-alike in the field is San Jose scale. San Jose scale is not a native. It's most likely been introduced from China, but it was first found in San Jose. It is circular, has that circular waxy coating with a very discrete nipple in the center. And it has one life cycle per year. What's unique about the San Jose scale and allows it to be differentiated from walnut scale is it has no discrete ridges on its outer body. And when I first started as a farm advisor, I remember hearing Bob Beatty give a talk about San Jose scale. And he said, the smooth body. Think of it as the city slicker. In other words, San Jose is a city, the city slicker. And that stuck with me. And I thought, I'm going to pass that phrase along because it really is excellent for remembering. So the walnut scale is actually a native scale, and it has a ridged body. And I've put arrows here to show where these ridges are. And it has a very scalloped, daisy-like armored coat on it. The walnut scale is actually native to North America. So I logged into one of those citizen science platforms, Discover Life, and I looked at where it's been reported in the United States. It's not just a problem in California. It is a problem on the East Coast, and there's a lot of entomological papers about it in North Carolina, Tennessee, etc. It has a wide host range. Now, it might not be one single concrete species throughout the United States, but no one's done a published genetic test on this, so it might be what we would call a species complex. However, Emily Sims, a former UC Cooperative Extension IPM advisor, and Danny Little, another former advisor, did some work collecting walnut scale throughout California. And they had some genetic tests done that showed that the population in California is only one species. So why are scales a problem? Well, generally things that are endemic, they emerge for reasons. Usually something happens that makes things that were always there become a problem. So if we look at the historical scale problems in California walnuts, we find that after the emergence of DDT for controlling other insects like codling moth, we had an increase in incidence in populations of other scales to the point that they became damaging to walnuts. And to quote this California Ag paper from 1959, practically all of the trouble with the soft scales on walnuts has developed since the advent of DDT that has interfered with the effective action of natural enemies. So in this situation, the advent of new products has affected the natural enemies of these scales, causing them to become a problem. And there's several papers on this topic. For example, here's another one specific to frosted scale on walnut. So this is not true of just scale. This is true of other pests and diseases. So disturbances or changes that occur in ecosystems may cause these problems, these diseases or pests to emerge and regress. Examples can be climate change, cultural practices, new genotypes coming in, as we release new cultivars, etc. And it can also be not just the introduction of new chemicals, but also the reduction of use of certain chemicals. So recently we've had things emerge that might be native, things like walnut twig beetle, thousand cankers disease, Botryopsvira has been in California, and these wood borer insects that I just looked at with Jalindra recently. So these have emerged as problems that we're actually now working on actively. So Emily Sims and Danny Lytle did a study, and I was a collaborator in this, managing sites that we looked at the phenology of walnut scale down in the southern San Joaquin Valley. And we had sites throughout the state in walnut orchards. And we looked at the phenology of the insect by wrapping double-sided sticky tape on scale-infested branches so that when the crawlers would emerge, they would get stuck on the tape. And we would then put these sticky tapes on paper and mail them to Emily, and her lab looked at the crawler emergence over time. We would take these data points every week. So this is a photo of what the crawlers looked like on the tape when you blow it up and when you magnify it. Historically, we considered the crawler emergence to occur in early May. And what this study did was elucidate that actually in some places the crawlers were coming out earlier. In my county, Tulare County, I had some sites where the crawlers emerged in late April. And this is the Contra Costa County site. And there was also April emergence there. So if you're timing your insecticide applications to first crawler emergence, you've got to consider where you are within the state. The other clear thing that comes out of this work is we see the two peaks of the crawler emergence. That's because there are two life cycles of this pest in California. So in this study, they looked at the peak activity of each life cycle and also when the first emergence was. And you'll notice that in several sites, we had April emergence. Also, we had this second emergence in later in the season with the peak activity around late August, we'll say early September. And in some sites, there was an overlap of that G1 and G2 population. Now, this is a diagram of the life cycle of walnut scale. This was put together by Danny Lytle when she was in Glen, Butte, and Tehama counties. The walnut scale overwinters as a second instar, okay? So we have this immature population that is available early in the season. The adult females and adult males are coming out or not coming out. The males emerge in late March, okay? And they are the only part of this insect that can fly. The only stage of the insect that flies are the adult males. The females will mature and become adult and fertile, and the males will mate. The crawlers emerge, as I said, in some sites as early as late April, that last week of April-ish. That first group of crawlers will then migrate, and they can colonize new areas, new twigs, the new growth, for example, the new shoots that were produced in that current season, and the life cycle will begin again. Now, one thing that I've communicated with Bob Van Steenwyk at UC Berkeley about this is that the female has 3 molts. It has a crawler and 2 instars, and then the adults. And the male is actually different. It has a crawler and 4 instars and adults. So there's 5 molts. The reason I wanted to understand this is because if you're using the insect growth regulators, they work by interrupting that molting process. They're like a juvenile hormone mimic. And so the opportunities for those IGRs to occur are at each molt. So for example, I put this in, if you do a delayed dormant application around March, when you think about what it would do as an IGR, it would be likely inhibiting the maturation of these nymphs into the adults, whether it's the adult female or the adult male. Now remember, that would have to occur before those males come out at late March to affect that male population. If we do an IGR application in late April or early May at the very first crawler emergence, this is going to affect downstream molts down here in that nymph population for the first cycle. It can also affect the G2, the second generation, and perhaps that overwintering population, depending on what's available at that time. Now, some growers have called me to say that they've been recommended to use an IGR application in January. There is no UC data on this. So if growers are doing a January application, realize that I'm not promoting that because I don't have UC conducted data. but there are studies that may be conducted by private industry. So I wanted to think through this to determine what happens in January if you do an IGR application. What could happen? Well, you can affect the development of this nymph population into adults, and perhaps you could even affect some of these molts that are occurring in the male population. So those are possible points that an IGR application in January could be targeting. So Bob Van Steenwyk did a lot of the studies on pesticide efficacy, insecticide efficacy for this insect. And they basically did this by spraying these branches and then coming back and counting the crawlers using that double-sided sticky tape technique that I'd mentioned earlier. These treatments in the study that I'm actually talking about were conducted in April. So it's almost like delayed dormant or a post-delayed dormant. And then at 6th of May at crawler emergence. So remember, if it was going on in early April, then it probably wasn't going to target the males because they come out by late May. So the walnut scale was then evaluated weekly from early May through June. And then the numbers of crawlers were counted. So I went through this to think about how these different insecticides work. And what are they doing and how are they targeting the scale? Because included in the study was Movento, which is systemic. It's moving up and down in the plant. And this would have been applied at crawler emergence. Assail is a neonicotinoid. It affects the insect with contact and ingestion. And so if we go down the list, down here at the bottom are the IGRs, like Cs and Centaur. And remember, they're affecting the molting. So this is the number of crawlers counted per day over that season. And you'll notice that Centaur, Assail, and Brigadier, Centaur, the crawler stage, and Cs were fairly effective at managing the scale populations. And if you look at them as the mean number of crawlers per day, here is your untreated check. And you can see which treatments were significant at reducing that crawler population. So we have these IGRs, and here is Movento. You'll notice it was not effective, okay? So Centaur was effective, Assail, Centaur, Brigadier, Cs were acceptable. And Assail at the low rate Movento and Sequoia were not different from the untreated. However, if we come back and look at how these populations performed during the following year, here's what were significant. And we include Movento in that assay in terms of its significance. And really, when you think about it, Movento is ambimobile. It's moving up and down in the plant. So it kind of makes sense that that would take a longer time to show an effect on the the insect population, because Movento would probably work better when you have that intimate relationship of the insect with the plant for a longer amount of time. So in short, Centaur and Cs are, they're the IGRs as a delayed dormant application, and Movento when applied at the crawler time. And that makes sense because you need the plant to be physiologically active and you want the insect to be physiologically active for the Movento to work. It provided excellent long-term walnut scale control 8 months after application. Brigadier, Sequoia, and Assail at the high rate provided acceptable control. So this is just the take-home message in terms of the efficacy of these insecticides. Now, if you find it a lot to put everything together from just one talk, I'd like to remind you that this information is available on the Fruit and Nut Research and Information Center website. So this is a UC hosted website. And the Walnut research reports are always available. They're archived in a database. So any study that was performed with funding from the California Walnut Board will be available in this database. And one thing I'd love to provide as a take-home message is that you don't want to count on Wikipedia for information, because the first thing I did when I Googled walnut scale to put this talk together was I got this Wikipedia thing and it puts the wrong order. It said Hemiptera, the true bugs. And so I really would like to emphasize, go to the UC resources because they should have the correct insect order. The key points that I'd like to make are that walnut scale is a pest in its own right. Changes in the ecosystem can impact these pests. It does exacerbate canker disease as per famous Michaelides' work and what we see in the field. Chemical control is a long-term strategy. It's not a one and done. You don't need to do multiple applications in a year because it can take time to see the results of an IGR application or even Movento. You might not see the results till the following year. So give it time and be patient. I wanted to take the opportunity to acknowledge a few people. First, Danny Lytle and Emily Sims. They were both advisors within the UC system. Danny's now at Oregon State University with the IR4 program, and Emily is now an entomologist with Sutera. Bob Van Steenwyck, obviously at Berkeley, has done probably the most amount of work on this pest, and famous Michaelides, professor at UC Davis in pathology. I also would like to mention Walt Bentley. He taught me a lot about this before he retired and even recently. He's an emeritus. And Steve Randall here locally is a PCA and crop consultant. And I'd like to thank him because he's the one that got me thinking a lot about walnut scale and the life cycle and when these IGRs would be effective and why. So with that, I'd like to take any questions.
Thank you, Elizabeth. Great presentation and right on time. First, we'll launch the poll questions, and then we'll do question and answer. There's at least one question. So,
okay.
Oh,
that's okay. We'll just do it this way. Oh, it looks like people are already answering. Look at that. We've all become Zoom geniuses out there. So if you've never done this before,
I can't see it, but okay.
That's
Managing
okay.
walnut scale in California walnut orchards. Walnut scale may predispose trees to canker diseases in the canopy. Well, that would be true. Right?
Okay. Oh, now everyone's going to get 100%. There we go.
Sorry.
That's okay, Elizabeth. Don't worry about it. No, we're doing really well. People are taking part. So I'm just going to go ahead and end the poll. Great job, everybody. As Elizabeth said, the answer is true. I'm going to go ahead and share the results.
Okay.
There we go. Do you see those, Elizabeth? You might not because you're a panelist.
I got it. Yeah, it's coming.
Perfect.
Through.
Okay, now what I'll do is I will share the next question. So these are also the questions that are on your DPR quiz. So no surprises there. Definitely take notes here, take part. Go ahead. Sorry, I need to launch it. Okay. Do you see the question, Elizabeth?
Yes, insect growth regulators, IGRs, work by 1, killing insects on the spot, 2, disrupting the molting process, or 3, making the plant tissues non-palatable to the insects.
Maybe just give them a couple seconds, maybe 10 or 20 seconds. Let everyone pile in their responses. Thank you so much for participating, everyone. I think these polls can be kind of fun. Nice learning experience. Looks like the polls are slowing down. Elizabeth, go ahead and give them the answer.
They work quite slowly in that they disrupt the molting process. So you have to wait for that molt to occur in order to see the result.
Excellent job, everyone.
Thank you. I'm sure
No
Steve
problem.
is embarrassed if he's watching this, that his photo's up there. Okay.
Not at all. Okay, so yeah, we do have some questions in the Q&A. Would you like
Okay.
to answer them live?
So I will read them out loud, Elizabeth. So the first question is, what is the best way to monitor for scale in walnuts?
Well, we don't have any thresholds for, treatment thresholds. So you would basically go out and just look for those scalloped scale bodies. And if you want to figure out when those crawlers emerge, it's really that sticky tape assay to get them stuck on the tape and basically look for them. You don't have to so much count them because it's not for research, but you just want to know when they actually emerge. From the data and your studies, what would you recommend as one spray per year in regards to timing? Oh, I really wouldn't feel comfortable because we don't have a complete suite of comparisons, but I would use the graphs from Bob Van Steenwyck's work. I think I would go with those IGRs. And I can't remember which one was used as a delayed dormant versus delayed dormant or crawler. So I would have to go back and look at that, look at which one for each timing.
Okay, we still have time and there are a few more questions. Next one is treatment required for survival walnut scale the following year?
You would have to go back and look again the following year. You can look and see if there's live female bodies underneath those scales. And that would be one thing. And, I've been wondering about this because the emergence of walnut scale is kind of a recent thing. And I've been curious if it might be related to a cutback in the use of Movento. Because remember, there was a lot of Movento use for nematicide as like a potential nematicide post-plant. And then when the walnut prices went down, I think, it's an expensive product. So maybe Movento use went down. And that kind of might have made those scale populations go up over time. I don't recommend using oil because I'm always concerned about oil killing walnuts. I don't know if any entomologists want to chip in on that or chime in.
And there's, you already alluded, I think, but there's a question about natural enemies. I think you said something about the fact that producers of products
the IGRs are supposed to be the better choice in terms of protecting the natural enemies. And I've seen like twice stabbed lady beetle out in the orchard, in some of the orchards down here. I didn't put pictures in.
So the next question was with respect to degree days and biofix. Is every other year a good general treatment?
I do not know. Do not know. I don't think we have data on that.
So there's a comment, I believe. And look for scale infestation on the wood of the tree. Right now is the best time to monitor walnut scale. So just direct observation.
That's a good point. Looking at the prunings is really a good point. We saw, Jalindra and I were just looking at wood borer beetles in trees, and we saw a tremendous amount of wood borer beetles, wood borer, walnut scale, and canker fungi. It looked like a whole complex within the orchard. And the walnut scale would be immature right now. So yeah, we had some pictures of them. Thank you for the comment.
Great. So
Okay.
no more questions. Thank you so much, Elizabeth.
Okay. Thank you so much.
And we'll move on to the next presentation. And can you please stop sharing, Elizabeth? Thank you.
Okay, how do I do this?
Should be at the top of your screen, like share screen.
Stop share. Okay. There we go. Thank you.